So we're finally going to go on and do something that you've heard your teachers talk to you for years about. We're going to take the ideas that you've learned and the ideas that you've learned and apply them to some concepts. So we understand about Mendel. Mendel introduced this idea of inheritance. We understand about chromosomes and how they divide during meiosis. When we start looking at chromosomal inheritance, we're going to understand how those factors that Mendel talked about are actually inherited on chromosomes, and how we can actually figure out the pattern of inheritance, and we're going to apply the patterns of inheritance. In lab and online, I've given you a lot of practice problems to do for this chapter, and I'm going to go through a few of them with you, but you're really going to have to take some time on your own and really learn how to apply the concepts, learn how to do inheritance problems and figure out patterns of inheritance. We can go on from Mendel, and again, Mendel, we talked about his work being completed in the 1860s. His work was largely undiscovered until the early 1920s. There was a guy by the name of Thomas Hunt Morgan who was researching inheritance. He discovered Mendel's work and continued his research using a fruit fly, Drosophila melanogaster, as his organism, and he came up with lots of really information. First off, he was able to look at the chromosomes of the fruit fly, and he realized that fruit flies have 4 chromosomes, an X and a Y chromosomes, and as well as 3 other chromosomes. And those chromosomes actually have different densities, and so you can actually see lines on them, and you can identify basically the characteristics of the different chromosomes. The nice thing about the fruit flies is they can regenerate in 2 weeks. It's easy to make hundreds of offspring from one cross. Easy to determine crosses. You basically, once the fruit flies hatch, you separate males from females, and then you can control the crosses. They're very easy to grow in a laboratory, and not too many people get excited when you kill a fruit fly. So he did things like introduce those fruit flies to mutagens, and those mutagens could do a couple things. He used chemicals and radiation, and they could damage chromosomes, or they could change chromosomes. And he noticed that when he introduced those mutagens, that actually there were changes in the characteristics of the fruit flies. He also noticed, for the most part, that they just killed the fruit flies, but there were some that survived, and those that survived would look different than the typical type. The typical type are called wild type. The wild type look like the flies that are on the bottom right-hand corner of the screen, those wild type, go backwards. Those wild type had red eyes. Learned a lot about just the ideas of inheritance, and again started applying things. So what he learned again is that Mendel was right, that alleles are carried on chromosomes, and we can start off by applying that by looking at a simple problem like this first problem, number 1 in the set that I gave you. So in cattle, horn condition is actually a recessive condition. In order to have horns, you have to have 2 recessive alleles. In this case, you're crossing a bull without horns, so that would be the hornless condition. It would have the dominant allele, according to what we've written down above. It's crossed with a cow with horns. It's a recessive condition that has to be homozygous recessive. They have 4 offspring. 1 is horned and 3 are hornless. And you need to determine the genotype of the bull and the cow. Now, the way we can go about that is we can take and write down some information. So we know that the bull without horns has the dominant allele, and he can either be homozygous dominant or heterozygous. So he can be big H or big H little h. And then we look at the offspring. He has 3 hornless offspring. His hornless offspring have received 2 recessive alleles, 2 little h's. And so if they received a little h, it's clear that they could receive it from the cow, but they had to receive that little h from the bull as well, and that lets us know that the bull is heterozygous and the cow is homozygous. Again, work on these on your own. Okay. Now, from that time we've learned some other concepts. We learned that the idea that Mendel initially disproved isn't necessarily right, so that there are some conditions where there are no dominant alleles. The case of flowers, in this case, impatiens and snapdragons, we found that when you cross the red flower with a white flower, you do in fact get the intermediate flower. And then when you take those intermediate flowers, the F1 generation, where they're all pink, and cross them, you end up with a 1 to 2 to 1 ratio of white to pink to red. Again, this is what happens when neither allele is dominant. Both alleles show up. Okay, again, I added some problems. Problem number 17 describes the cross between a red and white flowered plant. Now, in order to be red, if we choose the letter big R to describe red and little r to describe white, the red plant would be big R, the white flowered plant would be little r. Each of them would only give one of their alleles to the offspring, so the red one would give its big R, the white would give the little r, and so the genotype of all the offspring would be big R, little r. Again, because it's incomplete dominance, the phenotype for the heterozygous individuals is going to be pink. Okay, we also have conditions where sometimes there are multiple alleles. And an example of that is ABO blood type. And each of these blood types actually determines glycoproteins displayed on the cell surface. And also what's going on, the blood type alleles, A and B, are both dominant. Okay, if A is present, it's going to be expressed. If B is present, it's going to be expressed. Now, in this case, they're using the letter I to describe immunoglobulin, but you can actually do these crosses without using the letter I. But people who are blood type A can be AA or AO, the little I representing O. People who are type B can be BB, homozygous, or BO, heterozygous. People who are AB, okay, have to get both an A and a B, each on different alleles. And people who are type O get no allele for either A or B type, they're OO. And the reason this is important is our bodies actually produce antibodies to whatever glycoproteins we don't have. And so people who are type A, if you give them a transfusion with type B blood, antibodies to type B will actually cause the blood to clot and can potentially be deadly. People who are type O can't receive either A or B blood because they produce antibodies to A and B. And again, if they receive a transfusion, they could actually die from the transfusion if they got a transfusion from someone who was type A or B. Okay, so problem 12 in your problem set describes the 4 possible blood types. And again, I like the way that they use. They use the possible types. People who are OO are type O. People who are AA or AO are type A. BB or BO are type B. AB is type AB. So you have a young man who's type AB and his sister has type O blood. And in order for him to be AB, his genotype, if you go back up to here, his genotype has to be AB. Okay, in order for her to be type O, her genotype has to be OO. Okay, if they actually are true blood brother and sister, that could happen if one parent was AO and the other parent was BO. So in that way, both parents would be able to contribute the appropriate alleles to their offspring. Here's another one. A woman with type A blood marries a man with type O. They have 6 children, all of whom have type A blood. In that condition, you assume that she's she's homozygous. In all likelihood, after 6 kids, if she was heterozygous, if she was AO, you would have expected at least one O kid to show up. So in all probability, she is AA, and the parent who is type O is OO. Could they have a child with type O? Again, it's unlikely, but if mother is heterozygous, it is possible for them to have a kid who has type O. So let's go on and talk about sex chromosomes. We've already looked at the different chromosomes. Here's another karyotype. You need to know the term karyotype, which describes a picture of your chromosomes. And you can have a karyotype done with amniocentesis or with chorionic villi sampling when you're pregnant with a child. And analyze a child's chromosomes for damage or gender or whatever. And one of the things that comes up is if they have the X and Y chromosome, we can determine in advance that they're going to be male. If they have 2 X chromosomes, we can determine the gender of the child. They're going to be female. Where does that come from? Well, if females are XX, if you think about meiosis, they can only give an X-type chromosome to their offspring. Males, X and Y, can give either an X or a Y. And depending on whether the sperm carrying X or the sperm carrying Y fertilize an egg, that actually determines the gender of a child. We can talk about alleles that are located on the same chromosome as being linked, and this will make sense in a minute when we get back and look at sex again. So linked means that an allele is found on the same chromosome. If two alleles are linked, that means that the only way they're going to segregate, the only way they're going to, when meiosis occurs, the only way they're going to separate from each other is if crossing over occurs. And linked alleles that do not segregate are inherited together. We can actually look at the frequency of crossing over to determine how the physical distance between alleles. That's how the first chromosome maps were done. So this is an example of something that a cross that was done with fruit flies. They took some gray-bodied, long-winged flies and crossed them with some yellow-bodied, mini-winged flies. Okay, and they ended up with basically a 3-to-1 ratio, all either gray long or yellow mini. What was going on is because those chromosomes were linked on the same allele, they always stuck together. There was no 9:3:1 ratio like we saw on the previous dihybrid crosses. That's what you would expect. What they actually found when they did do crosses is they actually found that there was a slight difference from a 3-to-1 ratio, and they used that ratio to actually determine the location along the chromosome of those two different alleles. Now this is a nice picture because it describes human inheritance a little bit better. I've used, and I talk about eye color quite often. We usually think about dark pigmented eyes as being the wild type for humans, but we know that there's a lot of other eye colors between, as well as dark pigmented eyes, and those other eye colors are probably associated with mutations that have occurred over time. We can describe most human traits, though, as not having simple Mendelian inheritances. You're not either, you don't either have dark eyes or light eyes. There's lots of shades in between. In fact, most human traits have continuous variation. There's just not one type or another. We could generally describe traits like eye color, skin color, height and weight, and intelligence usually is being described by a bell curve. The picture that you're looking at is a picture of height. I believe those are students, both male and female students, and looking at the number of individuals of different heights. And they worked out really nicely to demonstrate a bell curve. Now again, sometimes the bell curves are a little bit skewed in one direction or another, but most human characteristics, because multiple alleles determine them, are best described by a bell curve. We can look at that to human inheritance. We can look at things like skin color and actually the possible distributions of skin color. What's interesting about human skin color is there's actually a link between environment and skin color. We know that part of that has to do with exposure to ultraviolet radiation, but we also know that people who have dark pigmentation in places where there's maximum ultraviolet radiation, so near the equator, people tend to have more pigmented skin. And that makes sense because ultraviolet radiation at the very least can actually destroy folic acid that's being carried in your veins. And so if you have very light skin and you're in an area where there's a lot of ultraviolet radiation, you may have less folic acid, which can lead to birth defects, which can lead to possible negative consequences for your offspring. We look at people in regions further away from the equator, and they tend to have lighter skin. And again, the hypothesis with that is that probably the reason that folks with lighter skin are more successful away from the equator may have something to do with vitamin D formation. Again, vitamin D is essential for calcium uptake. If you have limited exposure to the sun, it makes sense to maximize the amount of sunlight that's absorbed by the skin in order to produce vitamin D. So those are current hypotheses about why different locations find more frequent pigmentation distributions. Now what's interesting is in the days where we have vitamins that you can take over the counter, you could see where that in the long term we would anticipate that those changes that used to leave people from particular areas having certain skin pigmentation no longer provide the advantage that they once did. Okay, now let's go on and talk about sex linkage. Sex linkage is when a chromosome, when a mutation is actually found on an X or a Y chromosome. In this case, a wide-eyed mutation is found on the X chromosome. Males get an X and a Y. What happens is, because they only have one X chromosome, if there's a mutation on the X chromosome, it shows up. Let me repeat that again. Again, what's going on with these flies is the mutation for white eyes. If a wide-eyed male if a fly has white eyes, the white-eyed allele is found on the X chromosome. If a white-eyed male marries. If a white-eyed male reproduces with a normal red-eyed fruit fly, all of the F1 generation are going to have red eyes because they're going to get an X chromosome that contains the red allele from one parent. And then when you look at the F2 generation, the only place where the white eye characteristic is going to show up is going to be again in the male fruit flies, having only a single chromosome that has the gene with the X-linked trait makes it more likely for them to show up in the male offspring. Sex linkage is a little bit more important in humans. There's some sex-linked traits that we're very familiar with that men get much more often than women. Color blindness, hemophilia, which is a clotting disorder, and baldness are all found on the X chromosome, and again are more likely to happen in males than in females. Okay, so problem 10 is an example. In humans, hemophilia is a sex-linked condition, and normal clotting is dominant over the recessive characteristic of carrying hemophilia. Now, a woman who is carrying hemophilia would have an X chromosome with the dominant allele and a X chromosome with the recessive allele. With the disease, she's going to give one or the other to her sons. Okay, if her sons get her recessive allele, they're going to have hemophilia. If her daughters get her recessive allele, they're also going to get dad's X chromosome, and they're not going to have hemophilia. So what you would expect is, of the children that they have, all their daughters will not have the condition, and 50% of their sons would be expected to have the condition. This is a pedigree chart that describes hemophilia in the European royal family. Just to start off explaining things, the circles are females, the squares are males. If something is half shaded in, the person doesn't have the condition, but they're a carrier. And if it's fully shaded in, the person has the condition. You'll notice, just looking at the chart offhand, how many of the individuals that are shaded in actually have they're squares. There's no females that are showing up with the condition. Again, what we can look at there is we can look at that pedigree chart. They use Arabic numbers to indicate the individuals, Roman numerals to indicate the generations. And again, mating is indicated by a line connecting the offspring. You can actually do that with your family. I've seen people who actually have done that with looking at inheritance of things like alcoholism in their family, and you really can sort of follow down your family and look at the inheritance of different characteristics. It gives you a clue of whether a individual disease is being carried in your family. So there's a lot more things we need to add here. I'm going to give you a bunch of definitions. Let's start off with pleiotropy. Pleiotropy is defined as one mutation having many indirect effects, so it seems like more than one gene is involved. So this is the opposite of polygenic inheritance. With polygenic inheritance, many genes determine one trait. In this case, one gene determines many traits. And again, this is showing some of the things that are wrong. When you have sickle cell anemia, you have abnormally shaped red blood cells, but it leads to a whole host of effects in the individual who carries the disease. We can go on and talk about environmental effects. Now again, this is a Himalayan rabbit. Himalayan rabbits display pigmentation in their appendages where they're cooler. And so what they're showing here is if you actually were to strap an ice pack onto a Himalayan rabbit, you would expect that the ice pack would be enough that he could begin to produce pigmentation in areas where you wouldn't normally expect it to see that. Another example of that is Siamese cats with the coloring at the tips of their ears and their tail. Hydrangeas or snowball bushes, you'll notice that they come in either purple or pink. You can actually change the color of your hydrangea bush by changing the soil it is, by changing the pH of the soil. Epigenetics is the idea is that environment can turn on or off genes, and that's an interesting phenomenon, an interesting possibility, because we know some people, when exposed to toxins, can get very sick, and some others will not get as sick. Again, we think that many human diseases have multiple effects that cause them, things like cancer or heart disease, and it's a combination of the genes you have in the environment that actually determine whether or not you have that disease. Epistasis describes when one gene affects the expression of another. This is a nice example of incomplete penetrance. I love and I love living in the era that we do today, because you can Google the term polydactyly and you'll find pictures like that. There actually are people who are born with more than 5 digits on their hands or feet, and extra digits, extra numbers of toes, or different numbers of toes. And this is something that displays incomplete penetrance, which means that there are some people who carry this gene and will fully express an extra appendage. There are some people who don't have complete penetrance, and so they just have a little bump or something where the extra digit would have been. And again, these are nice pictures of the supernumerary digits. My mother was actually an X-ray technician, worked at Children's Hospital, and described seeing more than one case of the kids who were born with this little extra bump coming off the side of their hand. And what they did at that time in the 1950s is they would just tie a rubber band around it, and it would stop circulation, and eventually the extra digit would fall off and the kid would grow up with 5 digits. But it's a dominant condition, and you would expect that there's a chance that when those people have kids, that they might also display, have kids who have an extra appendage. Talk real quickly about the idea of mosaicism. Now, we know that men have a X chromosome and a Y chromosome, and women have 2 X chromosomes, so we actually have extra genetic material. We can't actually have all of our X chromosomes turned on at the same time. So one thing that happens with women is one of the 2 X chromosomes is turned off, and basically it remains condensed and forms this thing called a bar body. Okay, an inactive X chromosome would be found in every cell in a female, and it just kind of sits there and waits for division. It gets copied during divisions. That's all it's sitting there and doing. So one of our X chromosomes is active, one of them is not active. What that means is that women will sometimes display in different areas of their body, they'll display different characteristics. Makes a lot more sense when we think about it with fur color. You have to have 2 X chromosomes present to demonstrate this kind of fur color in a cat. Again, there's a nice picture here demonstrating what's going on with the fur color, the different X chromosomes being turned on and off in different parts of that cat's skin. I'm going to stop here, and then we'll talk about human genetic disorders real quickly, and that'll be the end of this chapter.